Great. So, as I said, today, the menu for today, it's mostly going to be spatial phenomena, let's say, not even spatial models. Let's talk about the spatial phenomena. So remember that we had, well, we said for the first three lectures, we said that the only thing we were going to assume we knew about these systems is that individuals reproduce. That's not totally true. We already saw when we derived the logistic model and when we derived these mean field approximations that we were assuming that the populations are perfectly mixed. And that requires something about the space. What we are saying, okay, they are perfectly well mixed and the space is not important. There is another cases in which the space is important. Examples were, for instance, if we want to study movement processes, then space is, of course, key. Even in cases where the individuals don't move, if we want to study plant communities or forests or like plant ecosystems, where the individuals don't move, then this assumption about the interactions breaks because a tree will only interact with the ones that has around it. There's no way to assume a well-mixed assumption in tree models, although people do it and they give insights. As a first approximation, sometimes it's a useful thing. It's way more incorrect than in cases where animals or in cases of organisms that move, but still people use it and it has given us a lot of insights of how these ecosystems work. So today what we're going to do, basically, we're going to relax this assumption of well mixing. And we're going to see which new things we can study in these more complex scenarios. So before we go to deriving the actual formalisms and the actual models that we're going to use, I want to show you some examples. I don't know why these slides are so slow. I want to give you an idea of the kind of spatial phenomena that we can study with these models when these slides allow me to do so. Okay, I don't have it here. Sorry, I have to share my screen in the computer. Sorry about that. Okay. I updated it. I updated the slides on Monday, I think, and I didn't make the transfer. But as you can imagine, processes that happen in space are going to be way more complex than the ones that don't have a spatial component. But they are also, of course, going to be much more pretty in terms of understanding them. So one example that we are going to study is what are called biological invasions. So biological invasions, and here I'm showing you one example, happen when new species, for instance, arrive to a new place. So now I think you can see my slides, right? And you can see the actual presentation, not the comments. Perfect. So the example that we are going to study first is an example that is the first example that was studied more using mathematical modeling. And it is the invasion of a species of rat that in English is called muskrat. The best translation I found to Portuguese is rato almiscarado, but I have no idea. I don't even know how to say this in Spanish. So let's say it's a rat. We are in a physics course, it's a rat. And the idea is that this species arrived to Europe from North America in 1905. And what you're seeing here in this figure 1 is a kind of contour map of how this initial population of rats spread through Europe. So here in the cross, we have the initial place where these individuals were found in 1905. And then basically, as time was going by, the author of this study, Escola, found that the species was increasing its range. It was increasing the area that it was occupying, and you see the years in each of the contours. At the end, by 1927, the area was much bigger. So what we want to study is how this spreading process is going to happen and how can we model it. This is the question that is going to drive us for the first hour, I'd say. Something that is more, it's very interesting, and we will touch on it at the end of the analysis of the model that we will develop, is that when this person measured the actual area of these contours, inside the area inside each of these contours, what they found is that the area grows linearly with time. And you can see here that the growth of the area is pretty linear. I mean, the fit is quite well. So these are the two goals that we are going to have. The first one is to understand how this spreading process may happen. How can we develop a mathematical formalism to study this spreading process? And the second one is whether and how we can get an explanation for this linear growth. This linear growth is basically telling us that the spreading should be somehow at a linear velocity. Sorry, this is not the area. This is the square root of the area. I was confused. So this is just one example. That was kind of the first one, is the one that you will probably see in most of the textbooks when you start studying biological invasions. Of course, there are tons of cases of biological invasions, and they are very, very important to understand because many times they threaten the ecosystem where these new species arrive. We have an ecosystem that, for instance, we can represent by these complex networks that we studied in the second lecture. The ecosystem is somehow stable. Everyone lives, they are happy with each other, or not so happy, but at least they coexist. And eventually we have a new agent that is arriving to that system. And that agent may have a very strong impact on the stability of the system and may create, for instance, may cause the extinction of a cascade of a species, which means that, for instance, it may overexploit one of the species that were at the bottom of this complex trophic web, and then this extinction in the bottom may cause a cascading extinction of the species that are above. So these invasions are really, really important to study. On a much smaller spatial scale, but to me also very pretty, these expansions or these range expansions have been studied more recently in microbial populations. So what I'm going to show you now is an experiment that I find extremely pretty and very interesting. There are tons of these experiments that you can find in many papers, where basically what they are doing is they are, I'm sorry if you cannot see the reference. Basically what they are doing, they are taking a petri dish. A petri dish is a dish of crystal, and they are inoculating a mix of two different species of microbia, let's say, two different microbes. In this case, I think they are even inoculating the same microbe, identical cells, and the only difference is that they are modifying them genetically in a way that some of them are seen as red in the microscope and other ones are seen as green in the microscope. They are identical. They can do that very easily to just manipulate the genes of these cells in a way that they express proteins that make us see these cells either as red, as green, or blue, the color that they want. That's kind of basic for microbiologists. For us, maybe fascinating, but for them, it's almost trivial, I'd say. So what they see is that in the very beginning of the experiment, you can see this plot here for 12 hours, the green and red are kind of well mixed. Everything is there in the initial drop. You put a drop, you can see, sorry, this line I think is 2 millimeters. So you can get an idea of how small these experiments are. And initially in the microscope, with the fluorescence that you see is basically green and red, and that leads to this yellow. But then as time goes by, these cells start reproducing, especially the ones in the border that are the ones that have, the ones that have a space to grow. And what they see is that this green now starts segregating in sectors. So now I'm creating, I see that in my experiment, there appears a red sector, a green sector, and eventually as the colony grows, this initially well-mixed population segregates in space and create different cones of different colors. So people have done tons of experiments on this system. They are measuring how these angles open as the population grows. They are even considering situations where there are mutations in these fronts, and this changes the form of the colony. There is a lot of experiments and a lot of theory trying to understand this. But this is a little bit, let's say, future work for us, or future reading for us. But I still wanted to show you these experiments. Of course, there are other spatial phenomena that may happen, and in these ones we will touch on them either later today towards the end, or tomorrow, or both, and are called spatial patterns. So spatial patterns are basically periodic distributions in a space of, yes, Davi.
I'm sorry, it's still about the microbes from before.
Yes. Mm-hmm.
I don't really understand why the different colors they group together, because from what I understand, like, aren't they identical to each other? How do the microbes like group themselves together with others of the same color?
Yes.
Can they see the colors, or just the researchers? That's my question.
Okay, yeah, that's a very good question, and I will clarify. So the idea is that you initially put cells that you have modified genetically in a way that when they express green or red colors. Okay, you will have reproduction now. Now, when one of these cells, you give food to these cells, which maybe I forgot to say, and that's what I didn't explain well, and these cells reproduce, they divide. And when a cell divides, it passes their their genetic material to their siblings, to their daughters. So then the daughters of a red cell are also red, and the daughters of a green cell are also green. So what is happening here is that every cell, when it divides, their daughters are close to her. So what you are observing is how, through different generations, the daughters stay close to their parents, and as you increase the radius of the colony, because you're expanding the disk, no? What you see is how the next generations allocate with respect to the parents. So that's why here, I don't know if you can see my mouse, can you?
Yeah.
Perfect. So here you have this very thin red line, and then you see that it opens a red cone. Why? Because when you initially had one red cell, then it divides, you have 2 red cells. Then you have 4. Then you have 8. You have a geometric expansion of that initial cell. I don't know if that makes sense now.
Yeah, I understand now. Thanks.
Great, perfect, thank you. So we will discuss an example that it's more familiar to me tomorrow, and I think it's going to be more clear because it doesn't have this radial geometry that it may be a little bit more confusing. But anyway, what I was saying you is that there are other, to me, very pretty example of a spatial phenomena that happen in biology that are called these spatial patterns. And these spatial patterns are basically spatial distributions of individuals, or in this case of a chemical substance, of a pigment in the skin of these animals, that are regular in a space. So here what you have is that, for instance, in this middle row, some animals, in this case 2 fishes and a leopard, they have their skins with some pretty pattern that people have tried to understand for many years. For instance, zebras are another example. These patterns happen in biology in at very different scales. They have very different characteristical sizes. So here, for instance, we have much tinier examples. The one in the left is the cornea, I think, of an insect. So really tiny nanometers. These are the eyes of a fly that you see. They also have a regular kind of structure. And of course, we even have in our fingers. What we have in our fingers, the fingerprints are also regular spatial patterns. But there is an example that maybe is less familiar to some of you guys because we cannot see it so frequently, and maybe also more fascinating because it covers huge spatial scales. So this bottom row, what we have are ecosystems that they self-organize over scales of kilometers, and sometimes many kilometers. So we don't have a spatial scale here, but we will touch on it later, probably tomorrow. I can tell you that these fairy circles here, this kind of pattern that you see here on the top, they form in the Namibian desert in Africa, although people have also found them in Australia and in other places. And I think the size of these rings is of the order of meters. So you can count many of these, and this covers kilometers, many kilometers. You can search for it in Google and you will be impressed by the scales of this self-organization process. This is another example where grass forms this kind of labyrinth that you can already think how similar this grass pattern is to this eye of an insect. It's kind of the structure is too similar for having, let's say, 12 orders of magnitude of difference in the characteristic time scale. And here what you have in the bottom, which is a little bit small, but we will discuss for a long time tomorrow about this example, is a regular structure, an aggregation process of muscles. Can you guys still see me? Okay. Sorry, I think my internet connection is not very well today, very good today. I will check every time. So these muscles, I think the scale of this picture is about 1.5 meters, more or less, but tomorrow we will see that this example is fascinating because the patterns go from 1-2 meters to kilometers. So, and we will give an explanation to this example. So these are the kind of phenomena that we want to study in these 2 lectures. So before we start developing the math, I want to know if you guys have any question about these examples or some curiosity that came to your mind.
Hello, Ricardo. I do have a question. How do you know that the same phenomena is that you can model the 3 things with the same phenomena, like tissue organization, animal codes, and landscapes are?
Yes, that's a very good question. So I'm not saying that you can model this with the same phenomena, although I can spoil a little bit and I can tell you that one of the things that we try to research is whether the same kind of processes are driving these aggregation patterns at these different scales. But that's open research. That's something that I am particularly interested in understanding, whether the type of interactions that drive the formation of these patterns of grass are also driving the formation of patterns between cells, for instance, because at the end, if you think as a physicist, you can think that there may be some universality or some kind of general theory to understand self-organization, but that's research. That's not something that we know. The only thing that I'm saying is that these are spatial phenomena that we can see, and what we are trying to see is to study the mathematical tools to study this. So if I don't know, you're a physicist or a mathematician or okay,
A
physicist. So basically what I'm going to claim today is that we can model this using partial differential equations. I'm not going to say that the phenomena that we are going to introduce in these partial differential equations is going to be the same. I'm saying that the mathematical tools are going to be the same, and we are going to see how can we recover these patterns. Okay. But yes, your question is a very legit research question. How do I know if they are the same? The question is, are they the same? That's still there. So to study these spatial processes, the first thing that we need to do is to relax, as I said, the well-mixed assumption. And to relax that well-mixed assumption, we are going to imagine that now our organisms, they live in a one-dimensional space. This can be very easily generalized to three-dimensional space, animals that move in 3 dimensions, in 2 dimensions. But to do this derivation, let's focus on a one-dimensional case that I think is going to be easier for everyone to follow. Let's assume that here we have our origin and we have a discrete space with some discretization L. And let's imagine that we have here, I'm going to remove red and black because I don't think they're very convenient for our background. Okay, so let's imagine that we have our happy individual here in the origin. Because now it is not well mixed, basically this particle is going to move in this space, or this individual is going to move in this space. Because my space is discrete, I am going to describe this movement by jumps, either to the right or to the left. Okay? And I will label, I will say that this particle moves to the left with some rate that I'm going to call lambda minus, and to the right with some rate that I'm going to call lambda plus. What do I mean by rate? Remember that a rate is a probability per unit time. This means that if I am observing my yellow animal here in the middle, every tiny time interval dt that I observe this animal, she has a probability lambda plus times dt of jumping to the right, and a probability lambda minus dt to jump to the left. And this lambda plus and lambda minus are always the same for jumps to the right, regardless of where we are. So we are assuming, if you want, that this tendency or this decision to jump to the right or to the left, in general, we are going to assume that lambda minus is different to lambda plus, that these differences come from some global thing of the environment or some intrinsic property of the individual. So we could very easily say that we have a lambda plus that depends on each of the positions. I don't know if I am being clear, but basically we can assume that, for instance, if I am here in the origin, I have lambda minus sub 0 or lambda plus sub 0, and if I am, if I am here, if I am in this 2L, I may have a lambda plus sub 2 and a lambda minus sub 2 that depends on the positions of the lattice. But we are not going to do that because, as you can imagine, the mathematical derivation of all the equations in this case is way more complex. You can find it probably in a statistical mechanics book, or even work it your own if you want, but it will not add many interesting things for the discussion that we want to keep today. So now I have this description of my system, and what do you guys think? Sorry, I'm going to say that this is X. So what do you think it's going to describe the movement or the dynamics of this system? How would you which function, or which I want to ask that in a way that I don't give hints. That's why it's hard to ask. So for instance, when we had this logistic growth, these populations that were divided, we said that the system would be described by the probability of having n particles at time t, because my system was stochastic. Now I think we all agree that our system is stochastic as well, because I have probabilities of jumping per unit time. But my question is, how do I describe this system in a way that I have the full information? Anyone has any intuition? Using the steps as a function, Nathan says. I kind of see where you go. Yes, number of steps. Okay, that's one example. For instance, if I give you every time a step, the number of steps that the particle gave to the right and the particle gave to the left. That's a way of describing the system, but that gives me information about one trajectory, right? Somehow, because I'm telling you in this realization, the animal moved 10 times to the right and 5 times to the left. If I want to have a full description of the system, the equivalent of this probability of having n particles that we had for the population growth process would be the probability of having my yellow friend at X, at position or at location X at time T. So if I give you this probability distribution function, this distribution of probabilities, then you have all the information. That's all you can tell me. You cannot tell me at time 10, at time t equal to 10, the probability is going to be here. A priori, you cannot say that. You can tell me with probability 0.3 is going to be there. With probability 0.5 is going to be there. So now that I have a probabilistic function to describe this system, what I can do is pretty much the same we did for the logistic model. I can write a master equation for this probability. So basically what I am going to do is to ask myself, what is the probability that my particle is at a position x at time t plus dt? So this is going to be equal, for those of you that have been practicing, this is going to be almost trivial, but this is, I'm sorry, I'm going to remove this 0 so I don't confuse anyone. This is going to be equal to the probability of being at x minus L at time t and jumping to the right, plus the probability of being an x at x plus L at time t and jumping to the left, plus the probability of being at x at time t and not jumping. So you can already notice from this very simple master, at this moment, this is not a master equation yet, but by this equation that I've written here, that this assumption of saying that the jump rates to the left and to the right are the same in all the locations, in all the nodes of my lattice, are going to simplify this equation a lot, because I can do this general thing of writing this for a general x. Something that is also simplifying my calculation a lot is that I am assuming that my particle only jumps a unit L, a distance L per unit time. That's also another assumption that you can very easily break it, and there are many models to study movement where the distribution, for instance, of the length that an individual jumps, let's say this is P of L and this is L, has different forms. For instance, there is one that is very, very common, that is called a power law distribution. So here this would be a log scale and this would be a log scale, that basically tells me that the animal will do a lot of jumps that are very short, but from time to time is going to make a very, very long jump. So these distributions, for those of you that want to read a little bit, are called Levy flights, and they've been thought to be very, very important for optimizing animal movement, animal searching, and navigation tasks in animals. There is a group in Natal, in Rio Grande do Norte, that is one of the world leaders in investigating this topic. So if you want to check it out, they've been doing very good job for many, many years. But going back to our very simple assumption where we have next neighbors jumps, the next thing that we have once, yes, sorry, Nicolas.
I want to ask something that is kind of basic, and I could have asked it in the first lecture, but I don't really understand why we have this, in this equation, we have like P of, I understand what each term means, but like you said, that each lambda is like probability over unit time, right? And so lambda times dt is the probability. I don't see why we need to multiply P of x minus L times that probability again.
Okay, yes. So so the idea is that you want to know, let's see, how can I explain this? Actually, maybe with this example, it's easy. You want to know at a time t plus dt, what is the probability that you are here? Okay? And now what you are going to do, you are going to count all the possible ways at which you can arrive here. The ways you can arrive here is if you are already here and you stay, if you are here and you jump, if you are here and you jump, if you are here and you jump, and if you are here and you jump. That we all agree, right? Okay. The probability. So imagine that that now, okay, let's simplify things a little bit. The probability of jumping in our case, the probability of jump, well, it's not important. It doesn't matter. So if you know that you are already here at time t, imagine that now you know that you are here at time t. Then you say, okay, the probability that I am here, that I will be here at time t plus dt, is just the probability that I jump. Because I know where I am at time t. So then I have two options. I stay, imagine that you flip a coin. If you get a head, you jump. If you get tails, you don't jump. So then the probability of being here at time t plus dt is 1/2. With 1/2 I jump, and with 1/2 I stay. What's the problem? The problem is that because we want to derive a general equation for the probability of being at a given place at a given time, we don't know where we are. So we basically need to count all the possible paths that will take me to that position. And my position at time t is probabilistic as well, because I don't know it. So when you are counting all the paths that take you to that final position, you need to consider all the previous places where you could be and the probabilities of you jumping there. And that's all I am doing. What I am doing is I don't know where I am. So the probability that I end up being here is the probability that I was here, and my coin tells me jump, plus the probability that I am here and my coin tells me jump, and so on and so forth. The simple case that we first described is when the probability of being here is 0, here is 0, and here is 0, and here is 1. So then I only have one path that takes me there. But in general, I have infinite, or I have as many as places I have. I don't know if that helped. Yes? Perfect.
It did help, thanks.
Perfect. That's very important. So I'm very happy that you asked. So what we can do next, we have this already this equation for the probability of being at location x at time t plus dt. So what we can do is the same we did to obtain the master equation. What we did was to expand on t. We assume that dt is very, very small, and we are going to expand in t. So basically, this is going to be the pxt plus the derivative of p of xt time. Well, in this case, let me see how can I do this, because there are two steps that I want to do at the same time. Okay. Let's don't expand it by now. What I do is px at t plus dt, and I do the same I did before. This goes to the right side, to the left side, and then I have all the dt's that can divide. Okay, nothing new for us. We already knew this. And this is equal to lambda plus px minus l dt plus lambda minus px plus l dt minus lambda plus lambda minus. Sorry, this happens when you write fast. This is at time t, I can move this up. Anyway, stay. So basically the same thing we did to obtain the master equation. So if you take a look at this equation, this is again a balance equation. It's a balance equation that is giving me the balance between the arrival to the position x, which are these positive terms, and the losses from the state x. And this is always the case for master equations, as we said. But what I'm going to do today, it's I'm going to go a bit further. We said that these master equations are very hard to solve because they are difference equations. We already saw yesterday that difference equations are tricky, even the simplest ones. And what I am going to do is instead of saying, okay, I'm going to calculate the mean value and I am going to be very, very humble, I'm going to be a little bit more ambitious, not too much, but a little bit. I am going to say, okay, if my jump distance L is very small, then I can somehow expand in Taylor series. I can do a Taylor expansion of this right side of my equation. And that's what I am going to do. So I'm going to say that if L is much smaller than 1, then the time derivative of P with respect to time is equal to lambda plus times, and here I have P of xt minus lambda plus is already outside, minus L derivative of pxt with respect to x plus lambda square over 2 and here I'm going to have pretty much the same, but with well, let's copy. With a different sign here because I have P of x plus and then finally what I have is the loss term that I don't touch because I cannot expand. Okay, so let's take this to the next page, which I think is going to be more helpful. And now you can see that I have a lot of things that will cancel each other here. So this and this are going to cancel with this. And basically what I have is sorry, I think I did something wrong. I don't know. So then what I have is I can reorder things and I get that lambda minus lambda plus L derivative of xt with respect to x plus lambda square over 2 lambda plus lambda minus second derivative of pxt with respect to x square. This is equal to the. So what did I gain by doing this expansion? Basically what I did was I went from an equation where jumps are a discrete variable of length L to an equation where my jumps are my displacement is a continuous variable. So I transform a difference equations into a partial differential equation. This expansion that I did here for a very, very simple case, and in a situation that is easier than usual, is an approximation to the master equation that is called the Fokker-Planck equation. The way you can understand this mathematically is that the Fokker-Planck equation is an expansion of the master equation where I need to assume that the displacement in my stochastic variable, the jumps in the value of my stochastic variable are very, very small. It's a diffusive approximation to a jump process. Okay, you can do that for every single stochastic process, for the growth rates, growth models that we studied in the beginning, for every single equation. Sometimes it will work better, sometimes it will work worse, but it's always possible to do it. So for this equation of movement of the probability of finding my particle at a given location x, now I can define both drift. I can define a drift velocity that will be it's lambda minus lambda plus L. And I can also define a diffusion coefficient that is going to be this lambda plus lambda minus over 2. So what are the first important things that we see about this velocity and this diffusion is that they have different dimensions. This, of course, has dimensions of velocity, but this diffusion coefficient has dimensions of square root of area divided by time. So they have different diffusions. They are going to give us different ways of transport, different ways of movement, if you want, and we will touch on that in a couple of minutes. So with these new terms, you can rewrite this Fokker-Planck equation in terms of constants that depend on the lattice. They depend on both on the discretization that you take and on the way that your particle jumps in that lattice. And that's not very convenient. It's not very elegant, but for us, it somehow will work, at least for today. Of course, there is a very interesting case that it is the case when we have many particles instead of having one particle. Imagine that instead of having one yellow ball in the beginning, I have a million of yellow balls in the beginning. So in that case, all particles, if I assume that all particles move independently to each other, that means that if yellow particle number 1 moves to the right, it doesn't introduce any bias in the movement of the yellow particle number 2, so they are independent realizations of the process. Then I can map this probability of finding a single particle at a position x to a density of particles at time t at position or location x. Is this mapping clear for everyone? Yes? Okay. So then basically I can, this Fokker-Planck equation for the probability of having, yes, Lucas?
You expanded the probability as considering the linear terms with respect to L, right? Because L is very, but why did you consider the L squared term in the diffusion coefficient?
Yes, that's a very good question. So I consider it for two reasons. The first one is because I wanted to show you how this, let me see. I want to show you how this randomness in the movement of the particles introduces this new different transport way of moving, this diffusion that we said. And the second one is because in most of the cases that we are going to study today, I am going to assume that lambda plus is equal to lambda minus. And in that case, the linear term, actually I was going to write it right now. In that case, the linear term is going to cancel, and I will be left only with the second order terms. Okay?
Okay.
Basically, the main idea, Lucas, is that I want to discuss with you the effects of randomness, of the movement being random on all these processes, and that's coded by this term. Basically, you can see that this linear term is basically some and let me know if I'm using like very technical words. This is some kind of deterministic drift in the movement. So this is some component of the movement that you will always see because the animal has more probability of jumping to the left than jumping to the right. So then you will see some linear constant velocity towards the left, because every time you decide where to move, you are giving some privilege to the left, for instance. So, and you can see it very clearly. If you basically take one of these lambdas to be equal to 0 and the other one to be equal to, I don't know, 1 or whatever you want, then in this case, for instance, if you take lambda plus equal to 0, lambda plus equal to 0, basically what you get is that the movement is basically deterministic towards the left. So anyway, so what I was saying is I am going to do these two assumptions. The first one is that I have a lot of particles that are independent, so I can map the probability to a density. And then I am going to consider that I don't have any preference towards the left or the right. I move with the same probability to both in both directions. So in that case, what I get is an equation that looks like this. And I want to know how many of you are familiar with this equation. How many of you have seen this equation before? You can raise your hands if you want. So that will be very helpful. Okay. Okay, not so many. Well, it's growing. Okay, not so many people. That's good. So we have the diffusion equation here. So this equation, what is giving me, and here you will allow me to just to make things a little bit more clear, you're going to allow me to use rho instead of p because I now want to talk about densities. And this is just notation, but I want you to keep in mind that now I don't have that single particle anymore. Now I have many particles that are going to move around. And what this rho is going to tell me is that at every time step, imagine that I fix t, I don't know, t equal to 10. I am going to take a picture of my lattice, and it's going to give me the number of particles that I will have in the center, the number of particles that I will have here, a kind of a histogram. Then I will take a picture and I will get a different histogram and so on and so forth. This is what rho is going to give me. And being more precise, because I am in this approximation of very, very small L, rho is going to be a continuous function in L. It's not going to be a histogram because remember, sorry, that I made this L to be very, very small. Anyway, so with this equation, in principle, it gives me some description of how my individuals are going to spread in space if they move randomly. But in the more general case that we want to study, usually we will have movement, and we will also have reproduction. And this thing that I'm going to do now is probably the less rigorous way, the less rigorous thing you will see, I don't know if in your lives, because we see many non-rigorous things in our lives, but probably in any physics or math course that you take. But what I am going to claim is that when movement and reproduction happen at the same time, I can modify my equation in a way that it looks kind of like this. Where I am having my reproduction term, and I have my movement or diffusion term, if you will. And because I took this equation out from my hands, I want to discuss it a little bit with you, the assumptions that this equation has. The first thing I want to say is that you can derive this equation formally by doing kind of these master equation things that we are doing for simple cases. It's just that the math is not so easy, and then I think well, we would probably not be able to do the full derivation here today. This equation basically what it is assuming is that if I have my lattice, and now you will allow me to instead of drawing my lattice as a series of marks, I'm going to divide my space in cells. Okay, I am going to say that this is x plus L, this is x plus, or being a little bit more rigorous, this is x plus dx, x plus 2 dx, x minus dx, and so on and so forth. What this equation is telling me is that in each of these x locations, I have one of these logistic growths. Okay, I have these spatial locations that are uncoupled, and I have logistic growth here. And then on top of that, I have this coupling between spatial locations that, that is telling me that these individuals that are reproducing at a given position, they jump with the same probability to the left or to the right. Okay, so if you with these assumptions, we get this kind of partial differential equation for the description of these populations, and this equation is usually seen in the literature as the Fisher-Kolmogorov equation. This equation was introduced in the 30s, 1937, I think, to describe how an allele is able to spread in a population of other genes. So basically, how when you have a population and you see with a very small frequency of some allele, or in the case that we want to, if we want to connect with the example that we discussed before, with if we put a little bit of rats, how they are able to spread in space. It has many other assumptions that you guys can probably think how to break. So for instance, it assumes that population dynamics happens in each of these locations independently from each other, so that individuals here do not compete or do not interact at all with individuals here. It also assumes that the birth rate, the reproduction rate is the same in every location. It assumes many, many things, but for us, it's going to be good enough toy model. So now the question that I want to discuss next is, what new phenomena do we have because of the diffusion term? Because if we take d equal to 0, we already know everything about this. Sorry, I was doing something very stupid. If we take d equal to 0, we already know what's the whole story about this model. Now the question is, what happens if d is different to 0, okay? And the first thing that we can do, what would be the first thing that you guys would do with this equation if you want to start analyzing it? We already saw it. So we have three parameters. Yes.
Maybe fixed points. I don't know if.
Yes, fixed points is is definitely one of the things we are going to do. Actually, it's the second thing we are going to do. I think. Yes, the second thing that we are going to do. But before we try to find the fixed points, I am giving some hint here. I'm saying that we have three parameters in this equation. So what I would suggest that we can do is we can try to reduce the number of parameters that we have in the model. So we can try to work in non-dimensional units to see whether I can write my equation in a way that is more user-friendly, more friendly for lazy people like me. So again, this is all based in intuition, and I invite you to try different non-dimensional units and different combinations and see where you go with each of them, because it's a very good exercise. The one that I am going to use, that once you see how the equation looks like, I'm pretty sure you guys will be convinced that this is a very convenient one, is the next one. I'm going to define a time scale I'm going to define a non-dimensional time that is measured in times of the growth rate, okay? I am going to define a non-dimensional density of individuals that is defined in units of the current capacity, which makes sense. These are very easy to identify typical units of my system. But now I need to non-dimensionalize the space, because now I have a space that is something that I didn't have before. So who wants to give some non-dimensional space, maybe guys that you are seeing here? Any idea? Okay, I'm going to write the easy one. The easy one is x tilde is equal to bx times
something with the square root of d.
Yes, excellent. Perfect. Why something with the square root of d? Because d is the characteristic spatial scale of my system, and it has units of a square of space. So it's something that I need to put here. So now what's the units of this? I'm sorry, you will allow me to complete this something by saying that is the inverse, because I'm looking for non-dimensions. But I guess the hard thing was provided by Nicholas. I'm just adding the final touch. So now this thing here has units of a square root of time, if I am correct. Yes, a square root of time. So basically what I think sounds reasonable is to grab the characteristic time scale of my problem and put it in a way that I get something non-dimensional. So if you guys calculate the dimensions of this product, of this operation, you will see that it has no dimensions. And this is the combination that I suggest we use. If you do the substitution, and I'm not going to do it, so you can practice, what you get is something that is going to help us a lot. And it is that this is how the Fisher-Kolmogorov equation will look like in terms of these non-dimensional variables and these non-dimensional parameters. Does this mean that my equation does not depend on the parameters? No, because the parameters are encoded in the non-dimensional units. So when you want to connect this to reality, you will probably need to take into account that every distance that you measure, every velocity that you measure is always related to the real units by this combination. So that's something that you have to take into account, okay? So now the next thing that I'm going to do is I am going to follow Amanda's advice. I like it a lot, and I'm going to say, what are the fixed points of my system? Now, I'm not saying that before it was hard, because I agree with many of you that the way this equation was written, it was not very hard to get the fixed points. But now it's even easier. I am going to assume that my solutions are uniform in space, okay? That everywhere I have the same value for u. So then the derivative with respect to space of this u is equal to 0, and then the fixed points are basically the same than in the case that I didn't have a space. So I have two fixed points, u star equal to 0 and u star equal to 1. So we can now do several things to this model, but now I'm going to guide the discussion a little bit. So we go to the new phenomena that we are looking for, and what I'm going to the point where I want to go is that we are going to see if how can we do this? Let's say capital A. We're going to ask if our model, or let's write this like a question. Does our model have, and I'm going to use a word that is the first time that I am going to use it, but we'll explain it in a bit. Does it have traveling waves? What do I mean by traveling wave? A traveling wave, I'm going to say that it is this kind of spreading solutions that I saw for the case of the muskrats. So I am going to say, what I am asking is if somehow, if I start, let's say my model, my equation, imagine guys that this is u and this is x. If at time equal to 0, I start with a solution that is something like this, what I am going to ask is if the evolution of this solution is such that this front is going to travel, with time. And this is going to be T2 prime, this is going to be T prime, and this is going to be T0 with T0 is more than T prime is more than T0. So what I want to know is if I start my solution with a non-uniform, I start my dynamics with a non-uniform solution in which a part of the system has individuals, let's say it's in 1 for simplicity, and another part of the system is in 0, has no individuals. I want to know what's going to happen. Now both my solutions are in the fixed point, points, right? I have here may have some continuous step to connect these solutions between fixed points. But what I want to know is how is this going to evolve? Is this going to make, is this the part with more than one, with individuals going to move and conquer territories that are empty? Is the other way around? What's going to happen? That's what I want to know. So the way I am going to look for these solutions is by using what is called an ansatz for this equation. So being not using jargon, which sometimes make things hard to understand, what I'm going to ask is if my solution u of xt can be written in a way where I have this translation symmetry, where c is some constant velocity. And that's the reason why I am talking about traveling waves, because I am going to test if my equation allows for wave-like solutions.
Shouldn't it be u of x minus ct?
Yes. If we're going to go somewhere, yes. Yes, perfect, Nicolas, thank you. And the next thing I can do is, well, I can say that x minus ct is equal to some other variable c, and I write it this way. So now the next thing that I'm going to do is something that probably many of you already did. I have done in some course of PDEs. If you studied, maybe also some in electromagnetism, depending what the curriculum is in your universities. But basically, I am going to do a change of variables in my equation. I'm going to calculate the derivative of u with respect to t. And this is going to be equal to the derivative of u with respect to c times the derivative of c with respect to t, and this is equal to minus c, the derivative of u with respect to z. Everyone follows this? This is the chain rule. I don't know if everyone is familiar with differential calculus. I am assuming everyone is. And then I do the same, the derivative of u with respect to x, and this, you guys can see that this is going to be equal to the derivative of u with respect to c. And if I do it twice, which is what I need, and using a notation that is going to be help me not making mistakes, this is something like this. So if I now put this if I now substitute the derivative with respect to t and x by their values with the new variable, then I get something like this. This is the derivative with respect to t, and this is equal to d, the second derivative of u with respect to c, plus u1 minus u. So this is a second order ordinary differential equation. This is what I gained by doing this change of variables, but I need I need boundary conditions, right? Because if not, I cannot integrate it. So what are the boundary conditions that, guys, you think I should be using? I'm going to give you a hint. The limit when c goes to minus infinity of uc, and the limit when c goes to plus infinity. So what should be the values of these limits? Now it's not zero. I don't buy zero anymore for both of them. Which are the limits? Sorry,
1 and 0.
yes, 1 and 0, Lucas, very well. Sorry, I think I confused you guys when I said I don't buy zero. What I meant is that usually when we integrate these equations, we say, okay, in the infinity, my fields should disappear or my solution should disappear. So it should be zero everywhere. What I was saying is I don't buy that argument because now I see and I know that if I have this type of solutions, when x goes to minus infinity, I go to 1, and when x goes to infinity, I go to 0. And what I have is this translation of a wave. So minus infinity should be 1, and plus infinity should be 0. All right, so here we are. But now we have a, well, I don't know if a problem, but we have a little bit of a situation here. The situation is that this system is second order, and we haven't analyzed any second order differential dynamical system so far in this course. And there is a way to solve this, that is the one that we are going to use. That I don't know if you guys have seen it, because sometimes it appears very early in some topics, which is that I am going to, yes,
something.
Sure, of course, every time.
I don't really understand why it should be 1, 0.
Yes.
I thought there was going to be something symmetric, like
Yeah.
spreading both sides, both ways.
Okay. So that depends pretty much on the initial pulse that you put, to be honest. So if you start with something like this, which I think is what you had in mind, then the expansion would be in this way. The only reason why it's 1 and 0, I'm going to assume 1 and 0, is because I am assuming that my initial solution is going to be something like this. Because it's going to make my calculations easier, and then for using symmetry arguments, you can go the other way around. But yes, depends on the initial condition, it will expand this way. Great. So the problem now is, let's write it here. Or the problem, or what we have identified, is that this is a second order. We have a second order derivative. How can you analyze this system? Anyone has an idea? No? I'm going to define a new variable that is going to be capital V, that is going to be equal to U prime. If U prime is equal to capital V, then V prime is equal to the U double prime, so to the second derivative of U. And if I do this change of variables, then what I have is that my system is now two-dimensional, but it's first order, and those are the systems that we learned how to analyze 2 days ago. So I can move a little bit further with this. The way you guys analyze this system, and then of course, I still need to write my my boundary conditions, which are the same in U. But the way you guys analyze this system is pretty much the same way we analyze the competitive Lotka-Volterra model. So we have a phase space that is VU. We can calculate the fixed points that the fixed points are going to be 0, 0, and 1, 0. And the only difference is that now V is the derivative of U. So when you plot your phase space, you have to, when you are thinking on the physical meaning of the variables, you have to consider that. You have to think that it's not that, I don't know, it's two species, it's just in one axis you are plotting the variable, and in the other one you're plotting the difference, the derivative of the variable. So once you guys have the fixed points, And I think now we are going to have a problem. So once you guys have the fixed points of the system, what's the next thing? The next thing that you can calculate, you can calculate the stability of those fixed points. I think because zero is not the boundary, the system goes. Yes, Rafael. The answer to that is what Nathan said. So Rafael asked, why not u prime at 0 equals 0 in that case? I think you mean why I take the boundary conditions in minus infinity and plus infinity is just because I'm considering minus infinity to plus infinity, as Nathan is saying in the chat. So once we have the fixed points, the next thing that we can calculate is the stability of those fixed points. And this, I invite you to do it in your houses when you have some time, because now we have all the analytical way to do that. We can calculate the Jacobian of the system, we can evaluate it in each of the fixed points, we can calculate the eigenvalues of the matrix, and we can do all the steps that we introduced yesterday in a very systematic way. If you want to play with this, you can also sketch the vector flow around each of the fixed points, and you can try to get an intuition. You can calculate the null lines, and you can do both the graphical analysis or the analytical thing. Both should agree. So if you calculate the stability of those fixed points, what you will see is that in 0, 0, we have two eigenvalues that I'm going to call lambda. I don't know if lambda plus and lambda minus is going to be a right choice. Okay, lambda 1 and lambda 2 that are going to be 1/2 minus c plus minus. And then around 1, 0, what we have is lambda 1, 2 is equal to 1/2 minus c plus minus. So now what I need to do is to see how are these eigenvalues, right? Depending on how these eigenvalues are, I will get a different stability for my fixed point. You can also write the Jacobian, you can calculate the trace and calculate the determinant and use the diagram that we built yesterday to go all the way to the point. But I wanted to give you the eigenvalues in case you want to calculate them and see how your results are. So if I call to this, let's say, green star, and this one, yellow star. Let's study first yellow star, green star, which is 0, 0. So who wants to tell me something about the eigenvalues of this on this fixed point? They are given by this expression here.
Both eigenvalues have a negative real part and they can have a complex part.
Yes. Everyone sees that? That depending on the value of C, I get what Rafael was saying? If C squared is larger than 4, then in this case, I have this number here is positive, so the square root of a positive number doesn't give me any imaginary part, and basically what I get is that this fixed point is an stable node. But if C squared is smaller than 4, this is basically a stable spiral, let's say. So yes. So if we want to use, to me it helps a lot because sometimes the names, well, I'm not very good to memorize names. So basically this is the behavior that you would get in these two different points, depending on the value of C, remember, depending on the value of C. For yellow star, we don't have that problem because this is, I have a positive sign here, so there is no way that this is negative. And basically what you guys will see is that this is always a saddle node. That, remember, was one of these points where I had an attractive, a stable direction and a stable one, and basically the trajectories never reached it. So let's see, to finish with this part, let's see what are the consequences. This is too thick. What are the consequences of this result? I'm going to put it very big. So this is 0, 0, 0, 0, and this is 1, 0. And this is V, and this is U. So which case do you guys want to study first? C squared larger than 4 or C squared smaller than 4? Okay, let's say smaller than 4. Let's start with a funny one. I am going to do a very hand-wavy explanation of what's happening in these cases. The proper way to represent the phase space and the phase portrait of this system is by calculating the eigenvectors. So you get the directions in which the saddle node is stable and the directions in which it's unstable, and you see whether my trajectories are going to be something like this here, or you know what I mean. You need to calculate the eigenvectors that we saw yesterday. But let's sketch it a little bit so we go to the discussion that we want to focus on, which we want to focus today. So we can let me imagine that this saddle node that actually I'm going to represent it like this, which is the way you will see it in books. So in books, when you see a fixed point that is stable, you usually see it filled. When it is unstable, you see it empty. And when it is a saddle node, you see it half empty and half filled. But that's just a convention. Wow, I put it this too big. Sorry. Okay, so let's imagine that I calculated my the eigenvectors of this fixed point, and I got that this is the direction in which these trajectories are leaving the fixed point. And now what I have is that I am saying that C squared is smaller than 4. So basically here I have a stable spiral, which means that that wherever happens, my solutions are going to close to the fixed point are going to be something like this. And not the prettiest spiral you've seen, but it works. Okay, and eventually, I don't know, you guys can play plot the phase portrait with the mathematical notebook I gave you. But basically, what we have are trajectories that would do something like this. If C squared is larger than 4, then the situation changes a little bit. Why the situation changes a little bit? Because close to the fixed point, I don't have oscillatory solutions anymore. I don't have a spiral. I don't have a spiral stable spiral here. So Instead, what I have is a stable node that is always attractive. Wait. So basically, this solution that was leaving from 1, 0 to 0, 0 now goes all the way to the fixed point. Right? The only thing that you guys need that is important for you to follow is that if I study the phase portrait close to the fixed points, in the case of 1, 0, my solutions are always going to leave in, let's say, in this direction. It doesn't matter a lot. And in this other fixed point, in one of the cases, I am going to have this spiral, but in the other, the solution may go all the way to the fixed point. And who can tell me what is a very, very important difference between these two things? What's the meaning of U? What is U? It's a
of the populations.
density. So what happens in these cases? Here and here. What's the difference?
So I guess in the second case, the density will reach directly to specific value, and the other it will oscillate in, I don't know if in space and time. I'm not sure.
Yes, it will oscillate in a space. And what happens when it oscillates? It takes negative values, right? It crosses the zero axis. And that's not very realistic for density. So the whole idea of this analysis is that if I have a velocity, a c square, a velocity of my traveling wave that is smaller than 4, my solutions are going to look something like this. Whereas if my velocity is larger, then my solutions are going to look like this. So the idea is that this Fisher-Kolmogorov equation allows for traveling waves, allows for propagation of these fronts only if c squared is larger than 4. Of course, there are many other further analyses that will allow you to calculate the exact velocity of the front depending on the velocity on the model parameters. It will allow you to even approximate the shape of the front to see if the front is smooth, if the front is sharp. And that's a whole thing of a whole field of research that many people study now. So depending on the nonlinearities, whether the front is sharp, the front is smooth, it has very important ecological implications, because you can imagine that if the front is very sharp, I don't know, maybe the invasion is with more individuals. If it's smoother, you have more individuals arriving, and there's a whole field of literature on this topic. The only thing I wanted to discuss with you was, first of all, the possible existence of these traveling waves. And second, which is what I want to do next, is what is the meaning of these waves? So what are the consequences? The consequences of these traveling waves. And we're going to do a very simple, I would say almost even naive calculation to understand the role of these waves. So what I want to discuss next, and I want to connect with this with the result that we saw that Scala found with the square root of the area as a function of time growing linearly in this invasion of muskrats, is I want to calculate the characteristic time scale of dispersal in the presence of traveling waves. So that time scale is very simple in the sense that we have a constant velocity c at which these waves are spreading, and therefore the the time scale of dispersal with waves that I'm going to call TW is basically some distance divided by velocity. Is this clear to everyone? The only thing that I'm calculating is what is the characteristic time that these traveling waves need to travel a distance L. And I am using the equation for movement at constant velocity. So it's not something very, very, very tricky. So this is what we get. Now I want to take the Fisher-Kolmogorov equation and I want to assume that I don't have creation of matter. I don't have reproduction. I only have diffusion. If you repeat all the analysis that we did to investigate the existence of traveling waves in this equation, you will see that this equation does not allow for traveling waves. In fact, you can solve this equation analytically. It's not very hard to do it. You can use, I don't know, guys, if you're familiar with them, but you can use separation of variables, where you basically assume that uxt is equal to x of x t of t, and you convert this into an eigenvalue vector, or you can even transform this equation into Fourier space and solve it as an ODE and then go back. There are many ways to do it. I'm not going to enter into that. But what you will see, and maybe you even already know it because it's one of the most well-known results, is that the solution to this diffusion equation has this form. So basically, the whole idea that I want to discuss with you is that when I don't have creation of matter, the mean by which I am transporting material, I'm dispersing these individuals, is very different to the constant velocity transport that I have when I create matter. And that's a very, very important difference. So who wants to tell me what would be the characteristic time scale for dispersal with only with diffusion? Anyone? So what are the units of diffusion? Diffusion, we said, was this. So the characteristic time scale in this case is going to be L squared divided by T, because now remember that the diffusion has units of a square here. And this is a very, again, this is a very
divided by D, right?
Yes, divided by D. Sorry, actually I think I did something. Yes. So now let's do a very small calculation. Let's say let's see what happens if I calculate the ratio between these two time scales. Everyone agrees so far? Yes. And now I have here this velocity that I have to write in the original parameters of the Fisher-Kolmogorov equation. right? So remember that I did all this analysis for the Fisher-Kolmogorov equation in terms of non-dimensional units that are defined by non-dimensionalized in space, non-dimensionalized in time, and then I go to this eigenvalue problem and I calculated this eigenvalue C. But this C, if I relate it to the original units of the problem, you will see that it relates to the growth rate and to the diffusion in this way, which makes sense. I mean, I don't have any velocity here and any actual velocity, right? What I have is that the interaction, the coupling between reproduction and diffusion is giving me a new way of dispersal that is at a constant velocity. But my individuals are diffusion. They are not moving at a constant velocity. And that's very important. I have, if you want to see it in this way, I have an emergent velocity as a result of the coupling between reproduction and diffusion. But I don't have my animals moving at a constant velocity of 3 kilometers per hour to the right. No, no, no. They are moving to the right and to the left. But when I couple that with reproduction, the effective transport process that I observe is at a constant velocity. And that's very, very important difference. So anyway, what I can do now is, and I will get rid of the 2 and I will use a proportional. Here I get this, and basically I get this. So let's take this to the next page. So who wants to discuss this result with me? Who wants to tell me something about this ratio? How do this ratio between time scales behaves as a function of D? It's proportional to this, right? So basically, we all agree that this ratio goes something like this. So what's happening here? When I have a biological system, for instance, imagine inside a cell, where I have to transport a lot of substances, a lot of molecules from one part of the cell to the other. So all the reactions happen in time and in form. How do you think that the diffusion constant inside a cell will be? Will it be large or will it be small? Will be small, no, in principle. We'll have tiny diffusion because the scales are very small. So what this result is telling me is that especially in systems where my diffusion is very low, by creating matter, by coupling the production of molecules, the production of individuals, the production of U to a normal diffusive transport, the gain in efficiency in the transport that I get is huge. It diverges actually with decreasing the diffusion. So basically, this the production of matter is a way to improve a lot the velocity and the efficiency of transport within a system, for instance. So you can think in terms of, I don't know, you can put numbers here like units of magnitude of characteristic size, characteristic production rates, and plot this, and you will see how much the transport of substances improves by producing the molecules. Of course, it will depend also on how fast you produce them, which makes sense because this new form of transporting is, as we said, coupling between production and diffusion. Okay. Does it make sense to everyone? I only see 3 faces, so if it doesn't make sense to someone, you guys can ask and we can discuss that further.
Ricardo,
Yes.
one of the assumptions we made in this model was that the the production was occurring throughout the space. So the
Yes.
particles would diffuse and they would reproduce, and so the production would be proportional to the density at that point.
Mhm.
In case of cells and other biological structures in which you have a center of production and a center of reception, does this reasoning also function?
Well, in that case, what you would have is a source, right? And then you have diffusion. But then probably in those cases, I mean, of course, this example that I put of the cell is a very, how do I say, it's a very big thinking example for a very toy model, no? Because if you are only if you're only producing in a place and then you need to transport, then the Fisher-Kolmogorov equation does not describe that process, right? Because you only have a source and then diffusion. Then the question is, you can calculate these transport processes depending on whether the production saturates, because imagine that, I don't know, depending on, and this is me elucubrating, speaking vaguely, you can think that, of course, this is going to depend a lot on the ratio between how fast you produce and how fast they diffuse, right? If diffusion is very small, then you will reach the saturation and then you will stop producing and you will get some stationary state. Or if it diffuses very fast, then I guess the production will be faster because you will always be in the exponential production phase. So my answer is that in those cases, you will probably need to study it carefully, but of course, this model doesn't apply. In this model, you need to have production everywhere. So something like the rat invasion that we were studying, whereas the rats invade, they stay there and they reproduce and they expand, or the microbes, for instance.
It makes sense then that in these invasions, what matters is the places where the animals they make residency, so they reproduce at that site. It makes a lot of sense on that.
Well, I mean, then of course there are people playing with this model in very, very different ways. We are going to see one of them probably at this time already tomorrow. But people assume things like the ones that you were thinking, like what happens if I write my Fisher-Kolmogorov equation. And for instance, the one that we're going to see tomorrow is what happens if, and you will guys allow me to write it this way. And I will use a diffusion now. I'm going to go back to units. So basically, what happens if instead of having this u minus u squared, I have some coupling with, remember the discretization of the space that we did? What happens if when I want for individuals that reproduce here, they also interact with individuals here and here? How things change in that case? Remember that we said one of the assumptions that I'm going to take to derive this equation is that reproduction happens independently in each of the cells, and then individuals just jump from one place to the other. What we are going to see tomorrow is what happens if reproduction in these places is enhanced by the presence of individuals here, but it is inhibited, is repressed by individuals far away. So for instance, imagine that, and I think this is a nice way to give an introduction without putting more math, because I think we are all tired of math. So imagine that in each of these cells, you have a plant, or you have many plants, pretty plants. If I want to see how these plants reproduce in this position, basically they will send seeds very close and they will put plants close to them, right? But in order to reproduce in that way, they probably need to take water from the soil, right? So a plant takes water from the soil, and if it has a lot of water, it reproduces. A very simplistic way of describing plant reproduction. Please, anyone with that knows biology, excuse me. But let's say that's going to be our picture. And what happens with the process by which plants take water? It happens that this process happens through the roots. And the roots are usually very long, longer than the plant itself. So it makes sense that although we see reproduction that happens locally in each of the cells, at the end of the day, when plants want to take water, they also have some interaction with far away individuals because they are fighting with each other through the roots. Or imagine, I don't know, a population of animals that are living in this lattice as well, and they go hunt farther than their cell. They go hunt, I don't know, to 4 cells in the neighborhood. So in that case, if there are rabbits in 4 cells, everyone that is at a given distance larger than the local, it's going to interact with each other. All those cases are represented by this kind of equation. This equation is, you can find it in the literature, like is the non-local Fisher-Kolmogorov equation. And it is called non-local because spatial interactions happen between, and maybe I should write x, t, x, t, xt, but the idea is that this quadratic term happens between individuals that are at position x and individuals that are within some omega domain around her. That's why it's called the non-local Fisher-Kolmogorov equation. It's spatially non-local. Those of you that have already taken more advanced quantum mechanics course, there is all this non-locality of quantum mechanics that is non-local in time and these kind of equations. In this case, this is the equation that we're going to study tomorrow for, let's say, 30 minutes before we jump to more research-like problems and more like a seminar discussion rather than a class, because well, I think Fridays are more for that, or for having fun doing non-science things, but let's stick to the plan. And what we're going to see is what kind of phenomena do we have in these equations. And I can already tell you from the introduction that you guys saw in the beginning that what we will find is that this equation has patterns. So when we have this kind of equation, when we look at u as a function of x, you will see that this equation has solutions that look like this, and that these patterns phrase in time. Just so you have an idea, the article where they introduced this, studied and analyzed this equation was published in 2003. So, well, now it's almost old. It's 18 years old. I hope there's no one that was born when after that paper was published, because you would leave me as an extremely old person. But what I want to tell you is that this is not anymore something that was done in the 20s. Or something that was done in the 40s or in the 19th century. This is something that was done in the 21st century, and it's an article that I will put in the Dropbox folder. It's an article that was written mostly, if I'm correct, by Argentinian researchers. So I think the main author is Miguel Fuentes. Marcelo Cooperman also is one of the authors, and it's yeah, it's 18 years old. So what we are with this very simple, what I want to say is that with these very simple tools and analysis that we're seeing here, we can see frontline research that was happening only 18 years ago, which in science, I can tell you is not so much. But that's going to be tomorrow. I promise I won't burn your brains with more math in 10 minutes. So if you guys have questions, you can ask them now.